I've been talking to students from Orange about this, and I'll tell everyone. The purpose today is to get a little context for the experiment you're going to do. If you are not careful, this whole experience this semester kind of turns into something where, yeah, plant stuff came in the mail, we planted them, and we measured them, and I don't really know why we did that. And then I had to write a paper and present it, and I don't really know why I did that. Well, that's the point for today, is so you know why we're doing this, so you can help get that context on where this fits in to a big problem, actually, a real-world problem. This isn't just data collection and everything else. It's got a lot of important information, and it actually directly applies to you. So again, your goal for today, take good notes. If you have questions, ask those questions. Don't leave here today feeling like you have no sort of understanding of what you're about to get into. This project, more than any other one you do in governor's school, kind of has a different feel to it. You're setting up an experiment where we don't really know what's going to happen. We make hypotheses, but we don't know actually what we're going to get. This isn't like a lab that a teacher sets up and we know what's supposed to work out. We have no clue. So again, it kind of differentiates it from the other projects you'll do. So anyway, take good notes. If you do that, writing the first part of your research paper is going to be a piece of cake. If you have questions for later, write them down. Talk about them in class. We'll talk about them here today, maybe. And we'll go over a little bit more of your project stuff in just a little bit. So I'll turn it over to Dr.
Okay. Good morning, everyone. We had breakfast this morning. Raise your hands. Okay, a lot of you did. That's good. That's a good thing, because they say that's the most important meal of the day. It was an easy thing, right? I mean, you went to the kitchen and either your parents had something ready or you fixed it yourself. That food in turn came from the grocery store. Either you or your parents went to Kroger or Food Line or whatever you have in your, in your towns, and you loaded up your shopping cart, you took it up and paid for it. Nothing in today's Western society really is easier, right? Simple, straightforward thing to feed yourselves. But the world's changing. Does anyone know what the current how many people live on the globe right now? 7.4 million. Yeah, right. Billion. Yeah, thank you. That's changing. The UN Food and Agriculture Organization estimates that by the middle of the century, the population will approach 10 billion. That's roughly a 30% increase. That means more mouths to feed on a globe in which we've pretty much hit up against the current capacity in terms of farmable land for food production. And this isn't really a matter of just making, 25 or 30% more corn, wheat, soybean, or whatever, because a lot of this population growth is occurring in nations with developing economies and rapidly expanding populations like China or India. They don't want to just eat rice. They want to have a protein-based, meat-based diet like they see their counterparts in the West, you and me, having. And so what that all adds up to is that, it's estimated that by the middle of the century, we're going to have to produce about twice as much food as we are right now. And to put that into perspective, okay, we're talking like 30 years. And we've come up again, and we're also approaching the limits of what we've been able to achieve with conventional breeding. Most of you guys probably heard about the Green Revolution that took place in the 1960s, right? That we were able to achieve a really big step forward in terms of our ability to produce food. That was done with conventional breeding using relatively simple, straightforward approaches. It was incredibly productive, but that low-hanging fruit, if you will, has been picked. And so what we're going to have to do now is get creative and become much better at working around the margins to increase our capacity to produce food safely and sustainably. Another challenge that we face is that, okay, not only are the demographics of the world changing, the climate's changing too. And so we have to think about sustainability. So would anyone care to take a guess at the amount of fresh water that's being invested now in food production? What percent? Any ideas on that? Somebody said more than 50. How many agree with that? Okay, all you guys who raised your hands are right. And it's by a pretty wide margin. It's currently estimated that about 70% of the world's fresh water goes to food production. Greenhouse gases are a factor too. It's estimated that roughly 30% of the greenhouse gases that our society currently produces are as a result of food production. So these are all parameters that we need to take into account as well to make sure that the Earth remains a habitable place 100 or 200 or 300 years from now. So this is the big picture. These are the problems that keep me and my colleagues awake at night. And so what I do is conduct research that occurs mainly at a fundamental level, but it's squarely aimed at addressing this big societal problem that I probably won't live to see, but you guys most definitely will, and your children certainly will as well. So that's what gets me up, gets me going, gets me to work every day. And what I want to do now is tell you a little bit about the work that I and PhD students who work in my group are doing to make a contribution, hopefully, to addressing the problem. The other thing that I want you guys to be thinking about and to understand really clearly is that, as Mr. B said, what you are going to be doing over the course of this project is original research. To design experiments yourself that bear on some of the larger questions that my group and I are interested in. And yes, we don't know what the results of those experiments are going to be. We don't know how it's going to be. So, yeah, you know, you might find this really intimidating, but I actually find it really exciting to, you know, to launch off on a course where you don't know where things are heading. I will tell you that if you're diligent and you pay close attention to the instructions that your teachers give you, the experiments will have good outcomes because they have all been through this system multiple times. They know what they're doing. This is, I guess, what, my 4th time through the cycle here, and the outcomes are always good. So I'm really looking forward to when we meet again in November to hear about your research. Okay, so what my lab is interested in are the effects that caused by microorganisms that cause disease on plants. And so what our contribution to this problem hopefully will be by the time I retire is that we've developed some new solutions to help reduce the amount of food that's lost to diseases. So I'm going to tell you a little bit about the work that we're doing towards that end. But one thing I want to stress is that I really want this to be a conversation. You know, I don't want it to be me just pure blabbing. I'm here at your disposal. Think of me as a resource that you can use to learn more about the things that I'm talking about. And that's going to continue throughout the semester as well. I'm available by mail. So as you begin entering your experiments and you begin encountering things that you're not sure about, reach out to me. I will respond as quickly as I can. And like I said, I'm very much looking forward to coming back here again and hearing about all the cool stuff that you've done. So let's pause here. Do you guys have any questions about the kind of big picture things that I was talking about a minute ago? Yeah, so the question is, what's going to be the role of so-called vertical farming, you know, where you're growing lots of things indoors on a relatively small footprint? And for sure, that's part of the solution. The really good work in this area is being done in the Netherlands. You guys know about Holland, right? It's a small country that basically exists almost entirely below sea level. The Dutch are really resourceful about food production, kind of leading the way in this area. For sure, that's going to help. That was a great question. Any others? Yeah. You're not jumping the gun at all. In fact, that segues perfectly into what I'm going to talk about. So yeah, I'll tell you more about that in just a minute. Who are the really bad actors? Yep. Any others though, before we get into that? Okay, so what do you guys think is happening with the plants in the bottom part of the picture? They're dead, or almost dead. They're sick. And so does anyone recognize the plant species? It's a crop. Think McDonald's. Potatoes. Yes. Yeah, so these are the potatoes, and somebody actually made the exact link to the disease that I was just about to describe. Who said potato famine? Bingo. Yep. Okay, so you answered your own question. This is one of the really bad actors. So the name of this disease is late blight of potato, because as you guys can see, these potatoes are blighted. And the name of the pathogen that causes this disease is Phytophthora infestans. Okay, so you guys know the saying, this is all Greek to me. Well, this really is Greek, but this word has a specific meaning. So phyto is plant, and thora is destroyer. So in Greek, this pathogen was named for its ability to destroy plants. And as you pointed out, this disease was the cause of the potato famine in Ireland. So this was a disease that struck in the middle of the 19th century, and it had really devastating effects. And the reason for this is that Irish society at that point in their history had grown to depend on the potato as a source of stable food. Now, this gets into, a pretty dark political history, but, at that point in time, the main job of most Irish farmers was to produce wheat. Now, they didn't get to eat that wheat. That wheat all got shipped off to England. Their main source of food was the potato, which had been introduced several decades before, and it had come from South America. And so the Irish adopted this, and the crop was extremely successful in Ireland's relatively cool and wet climate. And in fact, the adoption of the potato as a food source led the population in Ireland to explode in the first half of the 19th century. But then something really bad happened. A shipment of potatoes was transported from South America to Europe, and what was bad about this particular shipment was that it carried a large number of potatoes that were infected with this Phytophthora pathogen. And so that's how this disease was introduced from South America to Europe. And what then happened was that there was a stretch of several years where the climate in Ireland was really, really favorable for development and spread of this disease. And so what that led to then, massive disease across the country, the loss of this crop upon which the Irish absolutely depend for their daily food needs. The end result of that is estimated somewhere between 1 million and 2 million died of starvation, and at least an equal number were displaced. They immigrated because they simply couldn't survive in this country anymore, so they struck out to find better living elsewhere. And indeed, it's likely that somebody in this crowd owes their residence in the U.S. to that sort of immigration. I run into people who tell that story frequently, and that is likely true for some of you. Okay, so, this was a really horrific set of circumstances. but it wasn't an isolated incident in history. And indeed, even with our current technology, it's estimated that the current economic impact of this disease, even with all of the tools that we now have at our disposal, is $6.6 billion annually on a global basis. That's billion with a B. And to make that real, okay, if you could take that amount of money and buy potatoes at current market value, you could meet the caloric needs of the entire world for about 2.5 days. Any questions about this part? Okay, so I want to come back to the question that you posed about what are the bad actors. I've talked about one, but there are dozens, if not hundreds, of different species of pathogens that affect food production. And so in addition to this late blight disease, there are close relatives of this species that cause disease on soybean, crucifers, and even things like trees. So the next story that I want to tell you guys is about a relative of the late blight pathogen that emerged in California in the 1990s. Now, this particular pathogen doesn't attack our food supply, but it does attack hardwood trees that are foundational species in the ecosystems on the West Coast. And here you can see a picture of the sort of damage that this disease causes. Now, the pathogen that causes the potato disease is ancient. It's been around for a long time. We have a very clear idea of where it came from. In this particular case, the pathogen pretty much arrived out of nowhere, and we still don't have a clear understanding of how it originated. But what has become very clear is that this particular pathogen is not restricted to the sorts of species that it's causing disease on in California. It has what we call a broad host range. It can go to a lot of different hardwood trees, including many that are foundational species here on the East Coast. And so then the specter that this scenario raises is that sooner or later, this pathogen is likely to spread elsewhere. It's already begun to appear in Europe, and it's likely just a matter of time before it shows up on the East Coast and it begins doing damage. So what I've hopefully convinced you of is that there are a lot of bad actors in the world which impact our ability to produce food. And the metaphor that I like to use for organisms of this sort is I like to think of them as bioterrorists, because what they've been able to do is exploit plants as sources of food and shelter. And in order to be able to do that, they need to be able to sneak in and get past the plant's physical defenses and immune responses. And once they've established themselves inside the plant, they're then able to exploit these organisms as sources of food and shelter. And in particular, I want you guys to think about how microorganisms might use plants essentially as a petri plate to obtain the nutrients that the pathogens need to grow and multiply. Because how that occurs at a molecular level is one of the specific questions that my lab is interested in, and you guys will contribute to from the research that you're going to do as part of this project. Any questions? How am I doing? Can you guys hear me okay in the back? All right, good deal. Thanks. So what options do we currently have? You know, what do we do to take care of pathogens of this sort? And I tend to think of the options as existing in one of two categories. And the first, and one of the most widely used, is chemical control. basically pesticides that you spray on your crops to hopefully keep them disease-free. And this is how it works, right? Okay, you buy a chemical that's produced by Monsanto or Bayer or Syngenta or whoever that's targeted at the sort of disease problems that you might expect to face as you're growing your crop. And then, you apply those chemicals at intervals that are recommended by professionals who understand how best to apply these sorts of things. And this sort of mitigation often has really, really, really good results. But it comes at a cost. So this guy's sitting here on his tractor. He's likely going to be on it all day. He probably has other things relating to the functioning of his farm that he'd rather be doing. And so in the case, for example, of potato late blight, most commonly in disease-prone areas, you have to do these sorts of applications at least 10 or 15 times per growing cycle to keep the pathogen at bay. So this costs the farmer time. It costs the farmer money. There are also hidden costs. So a lot of these chemistries, aren't necessarily the best thing for the environment or for the humans that are associated with their application. Now, you can probably see that this guy is wearing protective clothing, and he also has a respirator. He also has his kid on the back who seems to be wearing a football helmet. And I guess the idea there is that if he falls off the back, well, maybe that'll help. But anyway, the point is that there are trade-offs associated with this sort of mitigation. Another big problem is that pathogens of this sort have a very high, what guys like me refer to as evolutionary potential. Put in real world terms, what this means is that if you keep applying these chemistries time and time again, season after season, then what's going to happen is that the pathogen is going to co-evolve. It's going to accumulate mutations that enable it to resist the effects of the chemistry. And over time, what will inevitably happen is that pesticide-resistant strains of Phytophthora or whatever you're trying to control will emerge and spread. And eventually, that particular chemical control will no longer be effective. Same goes for genes in the plant that provide for resistance. This can be another really, really powerful approach. But for pathogens like Phytophthora, they are in turn really, really good at evolving means to break that sort of resistance, and then those populations spread, and then you've got to deal with the same problem over again. So all of this is to say that we need to be really creative and have a lot of tools in the toolkit to deal with these sorts of problems. You guys have any questions or comments? I'm curious, do any of you come from farm families? Okay, yeah, some of you do. So you know what I'm talking about, right? Okay. So now I want to start talking about the research that we do in my lab. And the first thing that I want to talk about is this plant called Arabidopsis. Mark, you actually nailed it. Your pronunciation was perfect. And so the first thing that I want to do is explain why we use this Arabidopsis plant in our research. And simply put, it's because it comes down to logistics. It's a very convenient model system. I'm sure all of you guys have heard about, using the fruit fly or the rat as model systems for research that's aimed at human health. Arabidopsis is basically the fruit fly equivalent of the plant world. It has a lot of the same characteristics that fruit flies have that make them such great subjects for doing fundamental genetic research. And the analogy that I like to use for when I'm talking about model systems of this sort is that complex processes are best studied using simple systems. So imagine that you want to teach somebody how an internal combustion engine works, or you want to learn that yourself. Is that better done with a lawnmower or with a stock car? A lawnmower, obviously, because what that enables you to do is focus on the four-stroke engine and how the compression cycle and all of that works. Once you understand that, then it becomes a lot easier to understand how a much more complex system like a stock car functions. And the important thing to emphasize is that the basic principles of internal combustion are exactly the same in a lawnmower as they are in a stock car. And so that principle carries over to model systems and biological research too. Pretty much everything that we've learned studying Arabidopsis has broader implications for things like corn, wheat, potatoes, and soybean. Okay, so what is it about Arabidopsis that's so great? Well, the first thing is that it's very, very small. So here's a schematic of the plant. You're all going to be seeing exactly this with your own eyes in a couple of months. And just by way of scale, this is meant to illustrate how big the plant gets at full reproductive maturity. It's a pretty tiny little thing. And for guys like me, that's a big advantage, because one of the things that limits my ability to do research is the availability of growth space. So with Arabidopsis, I can look at hundreds or even thousands of plants in the area that exists within the basement of my research building where the growth chambers are. If I were doing this sort of research with corn or soybeans, then I could only do one growing cycle a year, and somehow I would have to come up with acres and acres of land to do it, which at Virginia Tech is a near impossibility, despite all of the great resources that we have. So that's a big deal. Another big deal is that it grows fast, much faster than any crop that you guys have heard of. What you will see again with your own eyes is that it can go from a seed to a seed producing plant in about 2 months. Again, that's a big deal because, research is a competitive enterprise. It's important that guys like me can do a lot of experiments in a relatively short period of time. So Arabidopsis is very advantageous in that regard. Now, another really nice aspect of Arabidopsis is that it's very easy to manipulate genetically. I can insert foreign genes or edit the genes that already exist in Arabidopsis using really, really simple procedures. I could take each one of you and teach you how to genetically engineer Arabidopsis in an afternoon, and it would work the first time you tried it and every other time after that. It's that simple. And that is an attribute that Arabidopsis is basically unique in. No other plant is as easy to manipulate genetically as is Arabidopsis. So those are the reasons I like to use it. Do you guys have any questions? Okay. So because we're interested in disease and disease losses, we need to have a pathogen to do experiments with as well. And so that's what I'm going to introduce you guys to now. And if you thought Arabidopsis was hard to pronounce, the name of this pathogen is Hyaloparanospora arabidopsidis. I call it HPA for short, okay? Now, the disease that it causes is downy mildew. And this picture does a really nice job of illustrating why this disease is named what it is. And the name comes from these white structures. And what these are basically reproductive structures for the pathogen. They're like little trees that are growing out of infected leaves that are going to make lots and lots of round spores that can get carried by wind or water to the next plant. That's how the pathogen propagates itself. Now, this particular species of pathogen is a natural pathogen of Arabidopsis. Arabidopsis doesn't just exist in the lab. You can find it all over the place in Northern Europe and even some places in Virginia. And wherever you find Arabidopsis, you're most likely to find HPA under environmental conditions where it's cool and wet. So we like this pathogen because it's really good at causing disease on Arabidopsis. Another nice aspect of it is that it's a close relative of these Phytophthora pathogens that we were talking about earlier. So the idea is that if we can learn things from this combination of Arabidopsis and HPA, there's a good chance that we can apply them directly to diseases like potato late blight. So in the last picture, I was showing you what happens on the outside of an infected leaf. This is a photograph that shows the inside of an infected leaf, and this was taken under a microscope. And the material that we were photographing was stained with a stain that highlights the pathogen structures. And so these are these dark threads. And so what's happening here is that the pathogen is about halfway through its life cycle. For the beginning stage, a spore lands on the outside of the leaf, and then it makes this thread-like structure, which we call a hypha. The hypha penetrates to the inside of the leaf, and it grows through the spaces that exist between plant cells. It just kind of snakes its way through the matrix. Now, as it's doing that, it makes interfaces with plant cells that are called haustoria. So here's a picture of what a haustorium looks like in a real leaf. And here's a schematic. So what's going on here is that as this hypha snakes in between plant cells, it makes these haustoria that punch a hole in the plant cell wall, and they then force their way inside the plant cell and exist there in kind of a stable arrangement. Now, the pathogen's not killing the plant cell at this point. This pathogen is a so-called biotroph. What do you guys think biotroph means? Yeah, that's a really good definition. Who said that? Well done. Okay, so bio basically means life, troph means feed. And so what this pathogen absolutely has to do is keep these host cells alive long enough for it to extract the nutrients that it needs to complete the life cycle, like I showed you in the slide before. So what's happening here is that this haustorium invades the plant cell. It makes that cell synthesize new membrane around the outside. And then what we think is going on is that this pathogen is also exporting its own proteins to the inside of plant cells to reprogram the plant cell to suppress the immune response and basically make those cells into food factories that the pathogen can then parasitize to take what it wants. This is a really important concept for you guys to understand why we're doing the experiments that we're doing. Do you have any questions about this? Yeah. Yeah, that's a really good question. How long does this all take? Okay, so if you think of this, so the spore landing on the plant, that'd be day 1. This picture was probably taken at about 0.5, 12 hours or so after we did the infection. This stage probably represents day 4 or day 5. And the end of the life cycle can occur within a week or 10 days after we do the initial infection. And the thing about this disease is, so you asked when does a pathogen kill the plant? It never really kills it. It keeps the plant alive through its whole life cycle. Remember, it has to do that. It's a biotroph. Dead cells are no good to it. It can't take its food from dead cells. So it keeps the plant alive, but by the time this happens, the plant is in a very, very diminished state. Its immune systems are weakened, its stature is reduced, its not growing well, and most likely it's going to be susceptible to other diseases that then will kill it. Things that wouldn't ordinarily be able to cause disease on the plant are able to do so because of the weakened state that arises from this disease. And you can think of it as being immune compromised and then subject to diseases that wouldn't ordinarily do it in. So that's a really good question. Are there any others? Yeah. Oh, right. Yeah. So this is, I think, 20x, not super high magnification. Yeah. And the leaf itself, you know, in terms of real size, is probably about the size of this fingernail or so. Yep. Other questions? Yeah. Oh, yeah, right. So where do we get the strains that we use for our lab experiments? You know, early on when I started this line of research, I developed a collaboration with a researcher in England who, as part of his program, was going out in the English countryside just collecting different isolates of this pathogen from natural pathogens of natural populations of Arabidopsis. And so he then mailed them to me, and we now just keep those strains going constantly in the lab as part of our research program. It's a good question. It's a real pain because this sort of research is regulated by the USDA. And, you know, the USDA, rightfully so, is concerned about the possibility of pathogens like this escaping out into the wild and having unintended consequences. So we have to spend a lot of time satisfying the regulatory agencies that we're doing this in a safe and contained manner. Any more questions? Okay, so you've met HPA, and let me go over this part one more time. What we consider the key aspect of HPA's strategy to parasitize plants is that it makes these Haustorium, which invade plant cells, and essentially turn them into food factories. And we think that this event is accompanied by the pathogen fundamentally reprogramming the genome of the plant to make that plant better able to feed the pathogen. Yeah, HPA makes the Haustorium. Okay, so this blue part comes from the pathogen. And these effector proteins here in orange, those come from the pathogen too. But these effector proteins then travel from the pathogen to the inside of the plant. And you can think of these proteins basically as molecular saboteurs. Their aim, you know, again, to use the bioterrorism analogy, is to enter the plant cell and somehow muck around with the control centers inside the plant cell that control things like the immune response or the plant's metabolism. And we think that a major part of that strategy is to reprogram the functionality of that plant cell so that it is now pumping out things like sugars and amino acids that the pathogen needs to feed itself. Another question. Yeah, right. That's a great question. So how does the plant adapt when it's under pressure by these sorts of disease-causing pathogens? And we think that the main adaptation that the plant can bring to bear is that all plants have really powerful immune systems, just like you or I do. And the basic molecular logic of the plant immune system is just like it is for human beings. The plant is making lots of proteins, like antibodies, whose job it is to detect when an invasion occurs. Those surveillance proteins, just like our own antibodies do, can directly bind to proteins or other molecules that the pathogen produces as part of its infection cycle. So you can think of that as a molecular recognition event. And if that recognition event occurs, then the plant is alerted to the presence of a pathogen, and it can turn on a really powerful immune response. Part of the immune response is for plant cells that have haustoria to commit genetically programmed cell suicide. And for a biotrophic pathogen that needs living plant cells to survive, that's kind of the ultimate weapon. So plants are far from defenseless in this, but in turn, that sort of selective pressure leads the pathogen to either discard the molecules that the plant is capable of recognizing or evolve new effector proteins that are better able to interfere with how the immune response is controlled. So this really is an arms race. And that's the perfect metaphor for the coevolution between plants and plant pathogens. And what's really striking about this coevolution is that it occurs really rapidly, almost in real time, but certainly over the course of years, which makes it relevant then to food production and plant breeding and things like that. Yeah.
Have you ever had a
Yes. Okay. So in this interaction, if we baby the plants, throughout the infection cycle, then it is possible to rescue plants that are then capable of producing seed. But, just to use seed production as a measure of fitness, which is kind of the ultimate criteria for evolutionary success, an infected plant probably produces maybe 10 or 20% of the amount of seeds that that an uninfected plant grown under the same conditions would. And if your fitness is diminished by 80% in the real world, then you're essentially an evolutionary dead end. Your lineage will not survive through the generations. Yeah.
How is the plant affected if they have to turn on that self-suicide?
Yeah, that's a great question. So when the system works the way it should, the suicide is confined to cells that are in the immediate vicinity of where the pathogen's invading. And so, what we're talking about then, if let's imagine that the immune response is triggered at this stage. What we're talking about are probably a dozen or so plant cells. So, even if all of these cells died, if you just looked at a leaf like that with a naked eye, you couldn't tell that anything had happened. Now, there are costs because in addition to this sort of local response, the plant also the infected region sends signals to the rest of the plant to let it know that trouble is potentially ahead. Now, that then causes the plant to direct some of its resources from normal growth and development to getting ready to defend itself, and that's not a good thing. So there is a cost. But overall, if the plant can recognize the pathogen in a timely manner, then it's going to win. It's game over for the pathogen. You guys are doing great. Yeah. Yeah, so that's another really good question. So how can we exploit this immune response of plants to essentially to control crop diseases, right? So literally thousands of researchers worldwide are working in various ways on that exact question. And our lab is also interested in that. We have projects that I'm not going to talk about today, which are designed to get at that. So for sure, that's a really promising avenue. But like I said, because these pathogens are so good at coevolution, we believe that we need to think of alternative means of genetic control. And so that's where the research that you guys are going to be contributing to comes in. And I'll talk more about that in a little while. Okay. All right, we already talked about obligate biotrophy. Now, the next thing that I want to talk about for this HPA pathogen is what we learned from sequencing its genome. So you guys have all heard of the Human Genome Project. Well, about 10 years ago, my group played a lead role in the Hyaloparanospora Arabidopsis Genome Project. And you didn't read about it in the headlines like you did the Human Genome Project. But we learned some really interesting things from sequencing the pathogen's whole genome and then comparing that genome sequence to the genome of pathogens that are related but have different lifestyles. And one of the most important things that we learned for the sake of the project that you guys are working on is that in this pathogen lineage, the pathogen has lost really important enzymes and transporters that enable it to take up inorganic nitrogen and sulfate. So, a potentially important source of food for a lot of different organisms are the chemicals nitrate and sulfate, which are found at pretty high abundance either in the soil or in organisms like you, me, or plants that pathogens want to reside in. And so for many pathogens, their favorite strategy to get these important elements is simply to absorb nitrate and sulfate from the environment and then metabolize those elements into forms that it can use for chemical building blocks. Well, as it turns out, these pathways over evolutionary time have become disabled in HPA and the pathogens that it is related to. And so for us, that provides a really important clue to understanding how the pathogen gets what it needs to eat from a plant host. So right away, then, we can rule out inorganic nitrogen and sulfur as important food sources. And what that then leaves us with is the likelihood that the pathogen is getting its nitrogen and its sulfur in organic forms that are synthesized by the plant as part of its normal metabolic processes. And so then the working hypothesis that guides the project that you guys are going to be helping us with is that amino acids and peptides, which are synthesized by the plant host, are an important source of nutrition for the pathogen. The second part of that hypothesis, which I spoke of earlier, is that this pathogen is actively exploiting the biosynthetic and transport machinery of the plant cells to feed itself. Again, this is a really important concept for understanding the experiments that we're going to be doing together. Do you guys have any questions about this? Take a minute and think about it and see if it makes sense. And if it doesn't, then let me know. Are you guys good with this?
No, like is it a mutation, and since it's parasite, it just doesn't matter because it can get the nutrients from the host, or is it actually it's just an energy efficiency thing, like it doesn't have to use energy to make its own?
Yeah, so it's both. It's a mutation that we think is, let me go back here. It's a mutation that we think is selectively advantageous because the pathways that provide for assimilation of nitrate and sulfate carry a big energy cost. It takes a lot of ATP to go from something like nitrate to something like glutamine or glutamate. And so what we think has happened is that this pathogen has become so good at coexisting with its host in a relatively stable state that it's able now to afford to throw away these energetically expensive pathways and simply rely on its host as a source of food. Put another way, it's become a very efficient freeloader. All good? Okay, so we have these hypotheses, but they didn't come out of thin air. There's evidence to support them, as there should be for any good hypothesis. And so what I'm going to do in the next couple of slides is show you some of the experimental evidence from my group, which supports what we're thinking about how the pathogen's exploiting the plant. Okay, so the first thing that I want to show you is this, this colorful diagram. And what I want you to focus on are the patterns of color that are evident in this diagram. So what each of these little bars of color represent are measures of activity of genes in the genome of Arabidopsis, the plant. So the activity of these genes were measured while the plant was under infection, either by an isolate of HPA that's very good at causing disease, or one that is not so good because it's recognized by the plant immune system. And so what the color patterns represent are that for every little bit of red, that represents a gene that has high activity during the experiment. And if the color is blue, then that means that these particular genes are not very active, or indeed their activity is diminished during the infection. Now, what sorts of genes are these? Well, in this graph here on the left, these are all of the genes in Arabidopsis that encode for transporters of amino acids. I'll talk more about these kind of proteins in a minute, but for now, the important thing to understand is that these proteins act as pumps that can move amino acids from the inside to the outside of a cell or vice versa. So in other words, they're the sorts of proteins that are depicted here as a pump that can take amino acids from the inside of a plant cell to the outside where the hostorium can suck them up. And so the main take-home point from this figure then is that, okay, this is a disease-resistant plant, this is a disease-susceptible plant. And what you can see from the red color is that a lot of the genes which encode these amino acid transporters are activated at a very high level during a successful infection by HPA. And so that then is consistent with the hypothesis that the pathogen is manipulating the plant's transport machinery to feed itself. So that's one lot of evidence. So my group is full of geneticists, and what geneticists like to do is test the function of particular genes by looking at mutants of that gene that disable the function of that gene. And then by examining the plants, we can make some inferences about what the role of that gene does when it's in a functional state. That's what you guys are going to be doing too. So the next experiment that I'm going to show you is where we took plant mutants in 4 different transporters of amino acids. We infected those mutants with the HPA pathogen, and then we measured the pathogen's ability to reproduce on those plants. And so this is a bar graph, and the important thing about the graph is our control. So on the left-hand side of these 2 graphs, that shows the degree to which the pathogen could reproduce in the wild-type plant. And what I want you guys to compare is the degree of reproduction that occurs in these 4 different mutants. These 2 and these 2 compared to the wild-type plants. So stare at that for a minute and tell me what you see. What's the difference? Yeah, it grows less good, right? Disease is reduced. And so that is another piece of evidence that is consistent with our hypothesis. And what we think is going on here is that in these plants that have mutations in these different amino acid transporters, this pumping capacity, if you will, is somewhat diminished. So there's less food available for the pathogen, so the pathogen can't grow as well as it could in a wild type plant. You guys good with that part? Does it make sense? Okay. So we have a lot of other evidence now which supports this hypothesis, but I showed you these two pieces to illustrate how we think about the sorts of experiments that we're doing to test our hypothesis. Now, let's talk a little bit more about amino acids. How are they synthesized in the plant? How do they move around? And so on. Okay, so, you know, the first point, which I think probably you guys know already, is that amino acids are very, very important for the proper functioning of every cell on this earth. So in the plant, amino acids are used as sources of carbon and nitrogen for metabolism. They can also serve as building blocks for so-called secondary metabolites, which have various important roles for plant ecology. And then, you know, the main role for amino acids that all of you guys have heard of before are as the building blocks of protein. Okay. Amino acids are fundamental. No organism can live without them. And so the way that amino acid metabolism works in the plant is that the amino acid synthesis takes place in the leaves. Okay, so these leaves are photosynthesizing. Carbon is being produced as a result of that. And the other thing that's going on is that the plant roots are taking up nitrate, which I mentioned earlier, and ammonium from the soil, and they're transporting those compounds up to the leaves. Okay, so here in the leaves then, the nitrate meets the carbon that's produced by photosynthesis, and those are brought together in amino acids. So the concept here is that the leaves in the plant are the source of amino acids. Now, the other thing that's happening here is that as these amino acids are being made in the leaf, they're also being transported either to the roots or the developing seeds where they're needed for growth. Okay? So you can think of the roots and the seeds as sinks for amino acid. And the way that they get there is through transport via the plant's vascular tissue. And the way that that transport works is that, okay, the amino acids are made inside these cells in the leaf, and they are then transported outside of those leaf cells into the space that exists between the leaf cells and the phloem, which will transport the amino acids to the sink tissues where they're needed. And that translocation from the inside of the plant cell to the outside, back to the inside of the phloem, is mediated by these transport proteins that I've been talking about. So think of them again as pumps that can take amino acids from one place to another, inside the cell to the outside, or from the outside of the cell to the inside. There are lots of different transporters in a normal plant that serve different functions in terms of which tissues they're active in and the direction that they mediate for the transport. Questions about this? Okay. And so this is the same picture of that plant illustrating how amino acids are transported from leaves to roots or to seeds. And what these three-letter acronyms represent are different genes in Arabidopsis that have been associated with amino acid transport in different parts of the plant. And so this is the point at which I introduce you guys to the gene that you guys are going to be studying in your experiments. The name of this gene is, the short name is AAP2. I call it APT2. This is just an acronym. The full name of the gene is amino acid permease 2. Okay, so this is one of roughly 30,000 genes in the Arabidopsis genome. We're very interested in this particular gene for a number of reasons. The first is that this gene has been pretty well studied at a biochemical level, and we know that the gene encodes a protein that is localized at the membrane of the plant cell, and it transports amino acids from the inside of the plant cells to the outside. We also know that this protein in a normal plant is active in the phloem tissue, and it plays this role that I was talking about earlier in transporting amino acids from source tissues such as leaves to sink tissue such as seeds or roots. Now, the other important thing that we've learned about APT2, go back here a few slides, is that this is one of those mutants that I was talking about earlier in which pathogen growth is diminished when this gene is rendered non-functional by a mutation. So by genetic criteria, this gene seems to be important for the pathogen's ability to grow well inside the plant. Okay? And it's not just HPA. What another group has shown is that this APT2 gene is also important for the growth of nematodes. Do any of you guys know what nematodes are? They're like little worms. They infest the soil, and they also can cause a lot of really bad plant diseases. Turns out, if you take an Arabidopsis plant that has a mutation in the APT2 gene, then the nematodes aren't able to grow as well in those plants as they are in a normal plant, just as is the case with HPA. So we think that this gene has a central role in the plant's ability to unwillingly support the growth of pathogens that are trying to cause disease. So we're really, really interested in learning more about this. And so we're putting a lot of energy in the lab into better understanding what this protein does during the disease cycle. So we're doing experiments designed to understand, okay, is this gene activated in cells that are actively being parasitized by the plant? Does the protein localize in the vicinity of pathogen feeding structures and things like this? And so in the long term, we'd like to understand whether this is one of the genes that the pathogen itself is actively reprogramming as part of its plant exploitation strategy. Now, everything that I've talked about so far is focused on basic research, understanding how things work. Our long-term dream, perhaps grandiose, we'll see, is that maybe genes which have a non-functional form of the APT2 protein could be utilized as resistance genes. So I showed you guys earlier that plants which have a mutation in APT2 are less well able to support the pathogen's disease cycle. What we'd like to know is, okay, if we go to potato or tomato and mutate the copy of APT2 that exists in the genomes of those plants, might those plants then be more resistant to diseases that are caused, for example, by the late blight pathogen? If so, that's really cool because that gives plant breeders a new tool to work with in producing plant varieties that are more resistant to this particular type of disease. But in order to think about whether that strategy really has legs, we need to be able to understand what other consequences a mutation of APT2 might have for the plant's ability to grow and develop in a normal fashion. So, and this is a really important point. So, I was just talking about how important amino acid transport is for normal plant growth and development. Is it possible that a mutation in this APT2 gene might have some bad side effects on the plant? Yes, it certainly could. And so that then leads us to the rationale for getting you guys involved. There are many plant processes, many aspects of plant growth and development that we're interested in regards to this APT2 mutation. But really, you know, this particular project is being worked on by basically one PhD student in my group of half a dozen or so. She's very resourceful. She's very hardworking. She has her hands absolutely full just with the experiments that I outlined here. So there are a lot of really interesting questions that this student does not have time to address. So does this mutation have other effects on the plant? How does it affect plant growth and seed yield? Does the mutation affect the plant's response to other sorts of environmental stresses besides pathogens? All of these questions are fundamentally important for this long-term vision of using mutations in the APT2 gene as a tool for better disease control. And these are the sorts of things then that we hope you guys can help us figure out. Any questions? Okay. So this is basically what I just said. You know, your role in this project then will be to design your experiments to better understand what sorts of consequences this mutation has on the plant's ability to grow, develop, and resist stresses of various sorts. One possibility that exists is that you may learn that plants with the APT2 mutation are much less fit than the wild plants are. That would be bad news from my point of view. Spoiler alert, classes before that have done experiments with APT2 have learned that the consequences are pretty minor, or in certain contexts, the mutation appears to be beneficial. But we need more data to help us really be confident that that is the case. And so then, the idea is that we'll be able to get some really valuable information from the work that you guys have done. Okay, so that's the end of my spiel. These are the main points. I'm not going to read them, but you guys read them, and if you have more questions, I'd love to take them. Yeah.
I was just wondering, because I noticed that you did AAP2, but there was also AAP2. Are those the same things, or is one the?
Oh, you probably saw a typo somewhere.
Oh, okay.
Yeah, so the name of the gene is AAP2. And so the typical shorthand for plant geneticists is that the acronym in uppercase represents the wild type gene, and the lowercase acronym is meant to designate the mutated form.
So if we find out that these mutations are good for incorporating them into, say, other plants like corn, do you think the public would accept it?
Well, you guys tell me. It's a good question. One really nice aspect, though, that bears on this is that, you know, what we are considering as a tool are naturally occurring alleles of AAP2 that can be isolated without direct genetic manipulation. There are ways that we can go into potato or tomato and screen for lines in which this gene has been naturally mutated, and then use those as tools for breeding. So this can be done with non-GMO approaches. Yeah, that's a really good question, and that gets at the heart of the experiments that we're interested in to understand whether the mutation in the gene affects its ability to grow and reproduce. So, you know, if let's say an APT2 mutation in potato has a negative effect on the potato plant's ability to produce tubers, then it's not going to be commercially viable. Yeah, so, again, to sum up what we know about APT2, a loss of function in APT2 seems to be beneficial from a disease control standpoint, but there may be trade-offs that affect other aspects of the plant that would rule out the possibility of using it as an allele for disease resistance. It all comes down to the bottom line. If you try to sell the farmer on a plant variety that has better disease resistance, but most likely reduced ability to produce the food that the farmer ultimately sells, then he's not going to buy it. He'll just say, "Oh no, I'll just carry on with spraying pesticides." And yeah. Yeah. So what a group of Arabidopsis geneticists did was to generate a collection of different Arabidopsis lines that have inactive copies of basically every Arabidopsis gene. And they did that by inserting a piece of foreign DNA into the middle of the gene so that the coding sequence gets disrupted and the proper protein can't be formed. So there are now roughly, I think, over 100,000 different lines of Arabidopsis. Each of those lines contains a mutation in one gene. And it's really easy for guys like me to go to a web interface and pick out the line of Arabidopsis that has a mutation in APT2 or which I'm interested in, and just order that up via the web. And it shows up in the mail a few days later.
Can you tell what her question was out loud?
Oh, yeah. So how did we get this inactive form of the APT2 gene in the first place? Where did it come from? It's a good question. And that's another advantage of doing research on Arabidopsis, is that we've got this tremendous collection of mutants that we can use for genetic experiments. Again, that's a resource that doesn't exist in any other plant. I saw other hands up. Were there other questions? Okay. Oh, no, here's one.
What would be the financial drawback of placing the old plants with ones with moved in?
Well, the answer, so the question is, what would be the financial drawbacks of using a plant with an inactive form of AP2? And the answer to that is that we don't know yet. And that, it depends on the effects that mutation might have on other traits of the plant, which is, that's kind of, like I said, that's unexplored territory, and you guys are going to help us figure it out. Worst case scenario, yeah, let's just draw an extreme case. The mutation renders the plant unable to make good roots because the roots aren't getting enough amino acids to fuel their own growth. The plants have bad roots, then they're not going to grow well. So what you would see is a really, most likely, finished, sickly looking plant or no plant at all when you sowed it out. That's an extreme case, right? That's probably not going to be the case though, because the plant has a lot of other amino acid transport genes that could potentially help compensate for the loss of APT2. That's the hopeful scenario. Yeah, it certainly can. So, there's material there for the arms race to continue, no doubt about it. And, I actually saw you looking carefully at the graph, and what you probably noticed was that, okay, although the degree of plant reproduction was diminished, it was still doing okay for itself, right? And that's probably because of those other transporters. So it's likely that an APT2 mutation by itself won't be enough for really viable control. We'll have to add other things in as well. Yeah. Could you say that again?
The mutation, have you ever tested whether it help stop diseases?
Yeah, so the question is, have we tested whether the mutation would help stop diseases, sorry. And yeah, so we've tested this for HPA, and like I said, the other group that's working on APT2 has tested it for nematodes, and it does have an effect. It's a partial effect, but it's an effect. Yep. Yeah, so, you know, when will we see the day where genetic manipulation is cost effective enough to address disease control problems? That's a really complicated question. In terms of actually producing the lines, that day is already here. You know, we have technology at our disposal that would let us go into something like potato or tomato and make a mutated copy of APT2 in a span of months, you know, with only one person working on the project. You know, that part of the process is very simple and straightforward and is going to get even easier as technologies are better refined. Where the big bottleneck lies is that plants which are genetically manipulated like this have to undergo a tremendous amount of scrutiny from 3 different regulatory agencies in the US. The Environmental Protection Agency, the US Department of Agriculture, and the Environmental Protection Agency. And all together, those ensure that, that genetically modified foods are safe for consumption and aren't going to have bad side effects from an environmental standpoint. That's all good, right? But the problem is that in order for an entity to gather the data that these regulatory agencies need to be satisfied that the product's safe, that costs tens of millions of dollars. And so what that then leads to is the reality that most GMO crops are produced by big companies like Monsanto because they're the only ones that can afford the investment that's necessary to have this sort of regulation done. And what that then means in turn is that in order for the plants to be profitable for a Monsanto, what they're basically focusing on are corn and soybean. The bad side effect of that is that that rules out a lot of so-called niche crops for which these sorts of modifications would be very valuable, particularly niche crops that are used by people in developing nations for whom food security is a big concern. But you know, it's at this point, it's simply unaffordable because of the cost of regulation. Yeah. So the question is, are there other genes with similar effects? Yes, there are. Yeah, so that's a great question. Why are we so excited about AAP2? The main reason is because it also has this effect on nematode diseases. But there are other AAP genes which seem to have an impact on HPA that we're also studying. Other questions? I think this is the best discussion I've had with the governor's school group. The other ones have been really good, but this one's been exceptional. I've really enjoyed this. That's all I have to say. I'll turn it back over to the MCs, and I think it's about time for some experimental design. Thank you guys for your attention. Absolutely.
All right. Can you guys hear me okay? I hate using mics. Comes from here. So I want to talk to you real quick about the project you guys are doing. If you already talked about this in your individual classrooms, great. I'll just go over a little bit of stuff that you may already know. First thing, if you go to the Governor's School website and you go to academics and current projects, you can get to the site this way right here. I just type in BRVGS Arabidopsis into Google and it's the first thing that pops up. But you have a project website that is a sadly underutilized resource during this project. And so I just want to walk you through it real quick before we start actually figuring out a little bit about what you guys are going to test. So on the homepage, you've got a countdown to when your papers are actually due, along with your presentation due date. So we did not put it down to like the individual second, but the minutes are a little stressful maybe. So that's how many days you got till the final thing is due. There's a document on here that outlines all of the due dates, everything like that. So please make sure you familiarize yourself with it. If you need any more information on some basic stuff about Arabidopsis, sounds like a good thing to write about in the introduction of your paper. We've got some resources on here. Feel free to use them. There's a ton of them out there. You guys will be setting up a website for each class where we keep track of our data. Where someone else from another school should be able to go to your website, see what you're seeing, see what you were testing, what were your hypotheses. All that stuff should be on there, and we'll actually be checking those as we go throughout the project. And if you didn't know this already, some of you may already know your groups. Some of you may not know that you're going to be split into further groups. Not everyone will be up here presenting their research on presentation day. Only some of you in the class will be doing that. Some of you will be making a scientific poster. Some of you will be going around and talking to the poster groups, and you'll be organizing the website and all the data during the project, so you won't have as much to do presentation day. Your guidelines for that are all on here, along with all of your rubrics, exactly how we really have a rubric in front of us, check off what you've done. So please actually look at the rubrics, okay? What I wanted to talk about right now is your research paper. So this is the biggest percentage of your overall grade. You're going to be working with people in your class, but everybody writes their own paper. That is an individual assignment. But again, you are working with the same data within your small group, so please keep that in mind. If you need any help on what to write, or just, I don't really know what my introduction should include, we've got things that you should include. I've had students in the past copy and paste this stuff into their paper and then delete it later. So they've got the guidelines right there. They don't have to keep going back and forth to the website. You can do that if you want. That's a good idea. But again, your word counts, what you should be including, that stuff is all starting right now. You've been given a huge amount of background information to use for your intro. You may have to do a little bit more research, but again, if you were taking good notes and paying attention today, it's pretty easy to write. One thing we don't know, like the last piece of the puzzle, I guess, that we have to figure out is what are you actually going to test, right? So we know that just having this mutant where we knock out the gene, we know the effects of doing that. But again, if you're paying attention, we don't necessarily know, does this mutant hold up to, I don't know, different conditions in the soil that maybe a normal plant would be fine with, but the mutant's not. We don't know that. So what we're going to do here in just a second, and if other teachers, if you have something to add, I'll give you a chance to say what I probably forgot. But we're going to be splitting into groups. And what we want to do before we leave here today is figure out what are we going to test. We don't want to have like 4 groups all doing the same variable. We start breaking out into our individual class groups here. What you need to think about is, okay, well, what affects plant growth? What normal things affect plant growth, okay? not like pouring soda into the water or something like that. Well, what naturally affects plant growth? What won't kill the plant if we expose it to it? Because if you just kill your plants, then we can't really tell, like, oh, look, the mutant responded the same way as a wild type plant. They died on the first day of the experiment. Like, that is not super helpful. So you need to think about something that's feasible. Can you actually test it? So for example, lots of people try to do things with light color. It is a really hard thing to control, and it affects things like temperature and all sorts of other stuff. So start thinking about what affects plant growth, specifically what affects maybe some of these sinks for amino acids like flowers and seeds. Can we think of something else that maybe would affect that? And again, something that we can actually test within our classroom. So that's what we're going to be branching off here and doing in just a second. Other teachers, am I forgetting something? Good, Ms. Dexter.
I just want to give you some pointers to make this an overall less painful process as you've experienced. Number one, you've been provided by a paper from Professor McDowell. Please do not get up and say things, well, we didn't expect these results and get in the paper. Those are the results we expected. You've got to do your background research. You have to read that paper. Even though it's intimidating and it's all got lots of work, read that paper. Two, you need to communicate with everyone in your class. So even though as maybe a pod of 4, you're running one trial, you have to be aware of what the other group's results are getting too, because the day of presentation, you're not presenting on your core, you're presenting on your class. The other big piece, just because you assign someone to collect data doesn't mean everyone isn't responsible for data. So if that person is absent, somebody else has to take the data. So you're all involved. So don't get into these, even though you may assign roles, that you get stuck in these roles, that you can't take a picture or you can't do this piece. You're all producing a product as a whole. You were all involved. And at the end, yes, part of your grade is like a group participation thing, but we are grading you as whole. And the last one with those rubrics, look at them, because there's nothing worse than you giving an A presentation and you forgot to put the citation page on the rubric, you just lost your 10 points on your presentation. There's very easy, quick ways to lose points. Look at that rubric, look at the rubric, look at the rubric. If you come to me and say, how am I graded in my classroom? I'm going to say, look at the rubric. Yeah, you may notice, but 20% of your grade is coming from the people who are. So it is in your best interest to work as a class on this, as a cohesive unit. And, you know, we do have personal points for the very rare times that something maybe isn't pulling their weight. But again, your grade for this project, a huge chunk of it is your individual effort, but not all of it. So that's something worth keeping in mind as we go through this today. Roughly 45% of your life.
Yeah.
55% is your class.
This is not to scare you. This is like really rarely a problem that we have to deal with. But again, some people are uncomfortable with the idea here that their grade might be dependent on somebody else's work. Science is a collaborative process, and just because we're associating numbers with it here doesn't change that at all. So just something to keep in mind. We ready to break out? Any other teachers? Anything else? Good, Ms. Alton. So again, teachers here, we're going to pick a couple students to go get those laptops. We can spread out a little bit throughout this room so you're not just locked into these chairs where we can't move around. I don't know. Dr. McDowell is still here. If you have an idea and you want to ask him, you want to run it by him, that's not a bad idea. So again, try to think what is feasible in our classrooms and what is actually applicable to these plants' lives.